The purpose of this lecture is to introduce the methods used to monitor and manipulate brain structure and function. We'll emphasize their utility in humans, but we need to appreciate that the discovery and the validation of the techniques required research with animals. Particular goals are to introduce these techniques used to look at the structure and function of the human brain, and then introduce the techniques used to manipulate the brain through the use of surgery or specialized disruption techniques. And we'll also review in a little bit more detail how drugs affect the brain and the mind. We will also introduce basic psychological experimental designs used to assess behavior and mental states during brain imaging or manipulation studies. It's been seen that the various methods used to study the brain can be arranged in a plot like this that plots space on the y-axis and time on the x-axis. So on the upper right, we see a box labeled lesion studies. One of the first ways that we became aware of the function of different parts of the brain was by studying the abnormalities of behavior consequent to damage to different parts of the brain. Some parts of the brain, damage to some parts of the brain caused you to be blind, other parts caused you not to walk, and so on. Now, these lesion studies occupy a protracted period of time. I mean, basically for the rest of the organism's life. And that's the time scale. On the spatial scale, they extend on the order of centimeters because natural brain lesions occupy several centimeters of brain tissue. If it's a stroke and things like that, or a gunshot wound, for example. Now then, moving to the left, at that large spatial scale of brains or gyrus, we have a method called PET, positron emission tomography. So this allows you to see the structure and the function at the level of a gyrus or larger, but its time is only on the order of minutes or longer. Moving further to the left, we see a green box labeled functional MRI. This is functional magnetic resonance imaging. So it can operate on a slightly faster time scale than positron emission tomography or PET, and it can operate on a somewhat smaller spatial scale as well, coming down into the order of a few millimeters. Moving to the left even further, we have a blue box labeled MEG, EEG, and ERP. This stands for magnetoencephalography, electroencephalography, and event-related potentials. So these are measures of the electrical or electromagnetic activity of the brain. And again, they're at a spatial scale on the order of the size of the brain or gyri and so on. But the temporal resolution is down on the order of milliseconds. It's a very rapid reflection of brain state, much faster than fMRI or PET. Now, embedded in the MEG is a technique called optical imaging, or EROS, event-related optical signal. And so this is a method closely related to fMRI, but it can operate somewhat faster, but it's at that larger spatial scale. Another technique, labeled TMS, stands for transcranial magnetic stimulation. This is a method that I'll show you that is used to manipulate the brain. It can operate on a shorter time scale, indeed one pulse at a time, for example, but it's got poor spatial resolution. It's on the order of a chunk of brain. Another technique labeled red, outlined in red, optical imaging. This is a method used in animal studies. It's not typically done in humans, but it uses the relationship between blood flow and oxygen level and nerve function to look at the distribution in space of the cerebral cortex of active and inactive neurons. Finally, lower, we see a box labeled multi-unit recording, and at the very bottom, at the 10 micron scale in space, extending from 1 millisecond up to 1 day, is single unit recording. This refers to the signal obtained from individual neuron or a collection of neurons from a probe placed inside the brain close to the cell body of one or a few neurons. So this is the highest spatial and temporal resolution available for functional studies of the brain. Typically, this is done in animal studies, but more and more often, especially in cases of treating Parkinson's disease or epilepsy, for example, it's possible to get single unit or multi-unit recording signals in humans as well. Let me first summarize the methods used to observe the structure of the brain. One of the first methods discovered is X-ray. So this shows an X-ray of a poor individual who tried to commit suicide by shooting himself in the head with a crossbow. And so you can see the structure of the skull, and you can see the location of the arrow in this poor gentleman's head. It's something like Phineas Gage that we saw in a previous lecture. And so this case was reported in a journal article in 2005. We find it interesting that the authors, or perhaps it was the editors, felt compelled to draw an arrow pointing at the arrow. X-ray shows us the structure of hard objects, like bone and arrows in people's heads. It doesn't show us much about the soft tissue. Now, an X-ray is a flat image. You can do it at different angles, and so if the patient lays with his head oriented vertically, we get an image on the left. If he lays on the side of his head, we can get an image like on the right. With the advancement of computer technology and other technology, it became possible to do more of a three-dimensional X-ray. And so this is referred to as computerized tomography. So imagine a set of X-rays taken at all angles around the head, and now you've got, from each two-dimensional image, the ability to make a three-dimensional representation of the object in the field of view. CT also has a bit more sensitivity, so it allows you to see the inner structure of the brain as well as the skull. So we can see in the image in the upper right a horizontal section of the brain. We don't know which is left and which is the right hemisphere because we're not told if we're using the radiology convention or just the natural left is left convention. We see the butterfly-shaped lateral ventricles in the image, and other areas where the sulci dip in. But there's not a lot more that one can see here, especially not with the trained eye. The major technique used today to look at the structure, the interior structure of the human brain, is known as magnetic resonance imaging. So the image shows a photograph of such a device. It's a very, very large magnet, very powerful magnet, into which the patient or the subject places the head. In this case, it could do MRIs of other parts of the body, of course. Through complex technology that we don't need to go into, it's understood, of course, very well, but we don't need to understand it beyond the fact that we get to see images like you see on the right. And that's almost what you'd see in an anatomical section of an actual human brain, if you just sliced it in a horizontal section as we saw before. So we can see the gray matter. It's the gray region in this monochrome photograph. And then the white matter is the whiter region. We saw these structures last time. Here's the caudate nucleus on the lateral aspect of the lateral ventricles. Here's the putamen. Here's part of the thalamus. And so MR, or structural MR, lets us see with high resolution the interior of the brain. And so with a scan technique like this, you could see if somebody has a cyst or a tumor or any other abnormality in the brain. It's interesting to note, MR studies like this are done commonly in a variety of research settings, and so more and more often we see individuals who have some abnormality of their brain with no symptoms whatsoever. They didn't know they had a problem. but often you'll see malformations of the blood vessels or other abscesses and so on that are completely asymptomatic, but when these are noticed, then a procedure has to take place to notify them of this potential problem. Now then, that's just showing the interior structure. Physicists are clever guys, and some of them realize that it's possible to look at the fiber pathways in the brain using magnetic resonance imaging. And the principle used is illustrated here. That's a long tube. Imagine it's an axon. And we're going to look at the flow, or consider the flow of water in the axon. Just like in any tube, it's more likely to be flowing longitudinally along the axis of the tube than to be flowing orthogonal or perpendicular to the axis of the tube. That fact alone, applied in an MR environment, affords images like this. This is what's known as diffusion tensor imaging. So there's faster diffusion parallel to axons than perpendicular to the axons. And as a result of that, one can image the large fiber pathways of the brain. So there's a set of 8 horizontal sections at different levels. On the upper left, we can see a section at a lower level in the head. So you see the 2 eyes toward the top of the section, more anteriorly. And with each successive section, we're going further and further up in the head. The red and the green and the blue that you see are fiber pathways running either front and back, left and right, or up and down, and the color is coding which pathway direction the fibers are going. And so this kind of diffusion tensor imaging allows investigators to see the pathway of major fiber tracts, and also to just analyze how much of the anisotropy or the diffusion takes. And so it turns out during development, the quality of the diffusion tensor imaging signal varies as the fiber pathways are gaining the structure of maturity. Those are some of the major methods used to study or image the structure of the human brain. Let me now review methods used to observe function. As I explained at the beginning, the highest resolution measure of the circuit function of the brain is recordings from single neurons in different parts of the brain. This is an illustration of the technique as it's applied in non-human primates. So we want to understand the function of the brain in an awake behaving state, and so the monkey has to be awake and behaving. So we can train them, as you would train your dog to do tricks with positive reinforcement of rewards, train them to do simple tasks like responding to a visual stimulus by releasing a bar. And each time it's done correctly, give the animal a juice reward to reinforce the behavior. Meanwhile, a recording probe or electrode is placed at a particular part in the brain, and the results of the signal are shown in the panel on the right. So we're imagining a situation where the animal is looking at a spot called the fixation point, and a light flash appears at another location. The panel on the bottom is showing the sequence of nerve impulses isolated from the probe in opposition to that neuron in response to the visual stimulus. So the visual stimulus occurs at the time indicated by the darker shading. Each of those vertical tick marks is showing the time of occurrence of a nerve impulse recorded from that neuron. Each row is a separate presentation of the stimulus to which the monkey responded and got a reward. At the bottom is a histogram counting the number of nerve impulses or nerve impulses occurring at each moment in time. And you can see in the period before the stimulus occurred, minus 400, minus 200 milliseconds, there was a sprinkling of nerve impulses, but not very many. After the visual stimulus was presented, the neuron fired a large number of nerve impulses transiently. When the stimulus disappeared, the number of nerve impulses was reduced again. So this neuron responded with nerve impulses to the presentation of the visual stimulus. This is a very elementary response, but neurophysiologists over decades now have worked up a very concrete understanding of many parts of the brain, or characterize the nature of the stimuli, or the action, or the emotion, and so on, for which neurons discharge action potentials. And we can learn more about how they're connecting and influencing each other. And it's at this level of analysis that we learn how the brain works, what mechanisms are at play. As I said, though, you can't do this systematically in humans. It's not ethical. It's possible to do in patients who have epilepsy and other diseases where these recordings inform the delivery of the therapy. In humans, though, it's possible to do non-invasive studies where you don't open the skull and get into the brain. One of the first non-invasive measures of brain function discovered in the early 1900s was the electroencephalogram, or the EEG. So we've learned that neurons are like little batteries. Electricity is fundamental to brain function, and it happens that when one puts a metal connection someplace on the head and measures a voltage from whatever point on the head to some reference, like the ears, then one sees a voltage. There's a small electrical potential that's measured. And the panel on the right is illustrating the nature of that EEG signal under different circumstances. When somebody's excited, the EEG is low amplitude and high frequency. It's changing, its polarity is changing very quickly. When the person's relaxed, the EEG becomes higher amplitude and slower, lower frequency. When they're drowsy, it becomes lower amplitude and then sometimes higher amplitude. When they're asleep, it has a different character. When they're in a deep sleep, it's much slower amplitude and even higher, slower frequency and higher amplitude. So the characteristics of the EEG relate to the behavioral state of the individual quite closely. The panel on the left is showing the location of contacts on the head, and you see they're labeled FPZ, FCZ, CZ, and so on. When investigators began doing EEG in humans, it became clear that across laboratories a convention needed to be established so that everybody would put the electrodes in the same place on the head to have comparable information. And so the system is referred to as the 10-20 system for the way that the electrodes are arranged, and the designations just describe where they're located. So F for frontal lobe, C for central, P for parietal or posterior, and so on. So in a law neuroscience setting, EEG data or quantitative analyses of it that I'll show you have become an important part of criminal law, for example. And so one thing you as a lawyer might want to make sure about is where were the electrodes placed? How many electrodes were on the head and where were they? So as I said, the EEG signal can be quantified in a variety of ways. One of the ways that's commonly used is to measure its frequency content. So this plot shows a diagram of an EEG signal, the voltage changing over time, and at the bottom is a plot of the amplitude as a function of the frequency, from 2 to 16 hertz, or cycles per second. And what we see is in this sample, there's a lot of signal at somewhere between 10 and 12 hertz, a little bit here between 8 and 10, and not much more elsewhere. Here's how you can think about it. This frequency analysis is something like decomposing the chord played on a piano. So if I play one key on a piano, that's one frequency, and so the frequency content would have one peak, one bend. If I play two keys, it will have two bars. 3 keys, 3 bars, and so on. Mathematical theorem says any slow wave like EEG can be decomposed into its constituent frequencies, and that's what quantitative EEG does. Decompose the EEG into different frequencies and relate the frequencies to, or identify the frequencies recorded at different locations on the brain or on the head. It's important to appreciate that while we know the EEG comes from the brain, more precise statements of localization are very difficult to make. Now then, another approach to analyzing the EEG is to create what's known as event-related potentials. So we have an individual with a cap now holding the electrodes in place. There's more electrodes and they're held in place by a cap. And the signals are amplified and we see an EEG signal on the upper right. We imagine that there's a stimulus being presented, and all of this is run by computer. We're able to do this because we have sophisticated computers. And so the computer knows both the EEG signal and the time when each stimulus is presented. And so in the upper right panel, we see S1, S2, S3, and so on. Those are the times of occurrence of the stimulus. We can do a process called averaging, in which we take the interval immediately after the stimulus, each successive stimulus presentation, we can take the EEG interval and line them up, synchronized on the stimulus, average them so that incidental noisy fluctuations up or down that have nothing to do with the stimulus average out, because it's equally likely to be noisy up as down, positive as negative. But the systematic variation that's resulting from the stimulus is what's left. And what's shown in the lower right is a visual event-related potential. So at time 0 is when the stimulus was presented. Before time 0, there's no net significant fluctuation. That's just noise. And then after the visual stimulus is presented, there's a sequence of negative going toward the top and positive going toward the bottom fluctuations. And these have labels, the NP80, the P1, N1, P2, N2, the first, second, third, negative going, first, second, third, positive going. Now, you'll notice in this plot that negative is up and positive is down. This is another convention in the field. And as an attorney dealing with information like this, it may be relevant to know if negative is plotted up or negative is plotted down. It could go either way. There's another wave out here, this third positive going peak that's off the edge of the display. We'll learn that this P3 component has been used to, or claimed to be useful for detecting whether somebody's lying about having some information. We can also construct color maps of the voltage or the frequency content recorded at different electrodes. And so this is a sequence of images from the back of a human head. The black dots are showing the locations of all the electrodes, and the color is intended to portray the voltage occurring at each slice of time, 0 to 25, 25 to 50, and so on, after the stimulus was presented. So this is another way of looking at the event-related potential. And what we see is after 75 or 100 milliseconds, there is a positive going wave over the right occipital lobe. And we would imagine that the stimulus was presented on the left side, activating this part of the brain. And then there's a positive signal over the top, and you can sort of imagine in your mind's eye a movie of this wave of voltage activation occurring over the surface of the skull, or the scalp. And indeed, you can make movies of these kinds of displays and see the spatial and temporal ordering of the voltages through time. It's important to appreciate, and this will become important in all of these brain imaging studies, that the color map used here was created by the individual making this display. And so, in the first place, the data were only recorded at the black points, but we have color in between the black points. And so the investigator used some algorithm to interpolate between the electrodes smoothly. The second point to make is the precise colors used are illustrated on the right. There's a mapping between color and voltage, but that mapping is arbitrary. And so it would be possible to create a map, for example, this is trivial, but you could create a map that was only blue to purple. So you would only see the blue and the purple, the negative going voltages, and not any of the positive going voltages. So people make these maps. These aren't photographs of the brain. Now, wherever there's electric current, there's a magnetic field. Wherever there's a magnetic field, there's an electric current. Electromagnetism is a fundamental law of physics. And so we can take advantage of that to record what's known as the magnetoencephalogram, which is recording the magnetic field associated with brain activity. This is requires very specialized equipment. The EEG can be recorded with very inexpensive equipment. Indeed, today it's possible to purchase an EEG, a one-channel EEG recorder for yourself off of Amazon to play with. But the MEG requires a very expensive, highly specialized piece of equipment, so it won't commonly be seen in a neuro law application. So we'll just move right on. Now, I talked to you in a previous lecture about the blood supply to the brain. And this on the left is showing an anatomical image showing or labeling the different arteries as they project to different parts of the cerebral cortex. And on the right is a pretty image reminding us of the dense blood supply to the brain. So the connection between nerve function and blood oxygen supply is at the basis of other techniques used to monitor brain function. The first we'll talk about, and the first that was developed, is positron emission tomography. Let's break down this complex phrase. So tomography you've seen before. Remember a computed tomography of the X-ray? So tomography is the idea of combining a bunch of two-dimensional images into a three-dimensional representation. Positron is a kind of radioactive subatomic particle that can be detected, and emission just means that it is emitted. So this technique is going to measure the emission of positrons in the brain coming from radioactive substances that are injected into the person's blood supply that gets flows through the brain. And so they're laying in a array of detectors. The positron is emitted through the radioactive decay, and it happens to have the physical nature of having two positrons go in opposite directions with equal velocities. So by having an array of detectors situated around the head, it's possible to know where that positron came from. And as a result of that complex physics, you can create images like this, which is a heat map showing the density of some radioactive tracer that was injected in this person. And so positron emission tomography can be done with radioactive oxygen or other substances like substances that bind to different neurotransmitters, neurotransmitter receptors. So in this image, we see it's a horizontal section. The front of the brain is toward the top, back toward the bottom. We don't know if left is left or left is right. That hasn't labeled, and so we have to be mute on that. The blue now is showing areas where there's less positron emission, and the hot colors, the red and the yellow, are showing areas where there's more positron emission. And that tends to be situated in the gray matter. So we see the cerebral cortex around the outside, and so this region, visual cortex, is particularly active. The ventricles in blue don't have a lot of positron emission because there's no neurons there to emit them. We see perhaps the putamen here and maybe part of the thalamus. So positron emission tomography allows you to see where certain radioactive substances are distributed in the brain. This can be oxygen or glucose and look at sort of whole brain utilization of energy consumption, or it can be particular molecules that will bind only to particular receptors. So for example, we can see how much of a dopamine receptor there is in different people in different parts of the brain. Another technique referred to as poor man's PET is single photon emission computed tomography, SPECT. This has some application in neurolaw, but it's becoming, I think, less common, and so we'll emphasize it less. Now, the most popular and widely used technique for understanding brain function today in humans is functional magnetic resonance imaging. So it's the same MR scanner that you get the structural scan for we saw before, but it's operated in a different way to look at the blood oxygen level dependent signal, or BOLD. And so we see an individual sitting in a scanner in the upper left slide, and the individual has some glasses on in which he may be viewing a movie, let's say, or some display, has a microphone in front of his mouth to report the responses, let's imagine. We don't know exactly what was being done in this experiment. And this is showing a blood vessel running through the brain. And here's the essential idea. There's layers of details, but the essential idea is as neurons consume, or as neurons operate and require more metabolism, they're going to require more oxygen to fuel that metabolism. And the blood vessels react to that need by providing more oxygen through the blood supply. And that can be picked up by the fMRI scanner in such a way that you can see the parts of the brain that are active or consuming more oxygen. The relationship between nerve function and this blood oxygen level dependent signal is complex and not entirely understood. So this is another important point to keep in mind, that when someone talks about fMRI activation, it doesn't necessarily mean that neurons are firing more nerve impulses. It may mean that they're being suppressed, because inhibition is also metabolically very costly. In any event, you get an image like that shown on the upper right, which is another horizontal section through a human brain. And we see the gray and white as the structural MR scan, and superimposed on it in red and yellow, and the red in the back is circled there. Those are regions with statistically significant blood oxygen level dependent signal. So there's a high degree of processing that goes into this. As I said, for the color map of voltage, this isn't a photograph of the brain's function. The red, its location, its extent, its intensity are all controlled by the user establishing the parameters to set up the display. Here's another way of showing the fMRI signal. We can show it in a slice on the upper right, or it's possible now with computer graphics to draw a three-dimensional rendering of the brain and then paint the brain activation patterns on it. So we see yellow and red, the hot colors meaning more blood oxygen utilization, and the blue colors portraying less blood oxygen utilization. And finally, so in the middle panel, we're showing the brain as it looks in real life with the sulci and gyri. As researchers began using these techniques, they realized it would be useful to inflate the brain and reveal the activation within the sulci. So imagine the brain as a balloon. If you blow it up, the sulci are going to become exposed as the brain expands. And that's what's plotted in the lower right, so that there's more areas of activation seen in the lower right, because those that were buried in the sulcus and not evident in the middle panel become plain to see. So in dealing with this as an attorney, it's important to recognize how the images were constructed, what the statistical criteria were, what the rules were for the color map. So in another session, we'll talk in detail about the limits of the method and the cautions that should be employed in interpreting it. Now then, functional magnetic resonance imaging studies are commonly done when people are performing certain tasks. And I need to tell you about two basic designs used in fMRI studies. On the left is shown a block design. And so on the y-axis is the MR signal, the bold signal, and on the x-axis is time. And we see the label AAA, and then rest, and then BBB, and AAA, and BBB, and rest. So A will refer to one task, and B will refer to another task, and rest refers to doing no task. And so we imagine that in task A, that's very demanding on the part of the brain where we're measuring this MR signal. So this is just from one area in the brain. And when they're doing the A task, it's more active. When they're at rest, it's less active. And when they're doing the B task, it's some intermediate demand. So we can look at the bold activation as a function of time as they're doing these tasks. And then we could do a subtraction, for example, of the state of the brain, the amount of bold in the A task, subtract the B task, and what we're left with is the parts of the brain that were needed to do A, but not B and not rest, for example. All right, now the problem with the block design is time passed, and the person may have got fatigued, or there could be changes that are incidental to doing task A and task B. So another approach is the event-related design shown on the right. Now, the upper right plots the relationship or the duration of the MR signal to one pulse of nerve activation, let's say. This is also referred to as the hemodynamic response. It's a slow process. So there's a stimulus A at time 0, and the MR signal takes something on the order of 5 seconds to become maximal, and then it's after another 10 or 15 seconds that it's righted itself, and there may even be a period of time when it has to compensate. So it may take 20 seconds to resume some baseline state. That's the hemodynamic response. But we believe that it occupies the same amount of time no matter what task is occurring. And so an assumption that it has the same time allows us then to create a design like this, which should remind you of the event-related potential in EEG. So we ask the subject to do task A, then B, then A, then B twice, then A, B, A, B. We interleave these tasks more rapidly in time than in the block design, so that the attentiveness of the subject and other extraneous variables are less of an issue. And then we can take the MR signal that's high and low and high and low and average it on the A event and on the B event and pull out the signal associated with doing A and doing B through time. That's the event-related design. There are more sophisticated approaches to this that we won't go into. Now, fMRI requires this large piece of equipment. It will be done typically in a hospital setting, although some of our colleagues have put an fMRI magnet in a tractor trailer and taken it to prisons and so on. But it's not the most portable and easiest to use device. This is a device that can be used by almost anybody. It's used for near-infrared optical imaging, or it's also known as near-infrared spectroscopy or event-related optical signals. It's a small box. It involves placing this collection of emitters and detectors on the head somewhere. And so what's shown on the upper right is its mode of action. So this emits infrared light. So it's something like if you've been to the doctor's office and they've put a pulse oximeter on your finger to measure your pulse. It's this red light that clips on and you hear the pulse. So the same idea is here. So the source emits the light. Some of it just bounces off the skin and so on, but some of it gets into the brain, not very deeply. And then the detectors on two sides detect what is reflected back out of this infrared light. And it turns out it has some relationship to the blood oxygen level state. And so it can be used to detect a signal, something like fMRI, more cheaply, more localized, but it allows you to find images like this. This are two sections, two horizontal sections through the visual cortex. The gray is showing an MR, structural MR signal. And on the left and the right are the optical imaging signals. On the left is for a stimulus that occupies the central 1 degree of the visual field, the width of your finger held at arm's length. And we see just a little bit of red on that shown by the crosshairs on the lower left. On the lower right is the same brain, but now with a stimulus occupying 2 finger widths. And we see a greater extent of activation because it's a bigger stimulus, occupying a greater extent of the cortex because that's where the visual field is represented. And so this near-infrared optical imaging is another useful technique to look at activation of the brain that happens to be near the surface, close to the skull. I want to introduce now the logic of double dissociation that is used to map function onto different brain regions. It can be used in functional imaging studies. The logic was developed and is portrayed here with brain lesions as well. So let's imagine that we damage one part of the brain, we'll call it region 1, and it results in a deficit in the performance of task A, but not task B. And so we can imagine region 1 to be visual cortex, and task A to be something that requires vision, where task B requires the sense of hearing. All right? And so on the lower left, we see the performance on task A and task B following damage to region 1. And so task A is done only 20% correctly following damage to region 1, but task B is done 60 or 70% correctly. Now then, damage to region 2 causes a different pattern. So damage to region 2 causes a deficit in performance of task B, but not task A. So in a trivial sense, we could say that damage, region 2 is auditory cortex. So now you can see perfectly well, but you can't hear. So you can do the visual task, but not the auditory task. All right, so that's the logic of double dissociation. And then when we apply brain activation to it, we see when the person's doing task A, we see more activation in region 1 than in region 2. And when they do task B, we see more activation in region 2 than in region 1. And if we see a collection of data like this, then we recognize that the two regions subserve or support the two different tasks. Key to applying this logic, though, is identifying the different regions discretely and identifying the tasks discretely. And this can become complex. Let me now talk about various approaches to manipulating the brain, some accidental and some purposeful. So accidental manipulations of the brain would result in damage or lesions, perhaps through stroke or cerebrovascular accident or some other insult. So this is illustrating horizontal brain sections through a sequence of individuals, top to bottom, 1, 2, 3, 4, 5, 6, 7, 8 individuals who all have damage somewhere in the frontal lobe, all right? And at the bottom is, so we have an MR image at different levels of the brain from near the center to the top, from left to right. And the red is illustrating for each individual where the damage is. Okay? Now, each of the sections, brain sections from top to bottom, it's the same brain. It's a standard brain onto which the researchers are mapping where the damage was in each individual. Because it's a common brain, they can be averaged in space. So at the bottom is the structural plot of the brain, but now the colors are showing the fraction of individuals with damage to that particular region. So most people had damage in this central region where I'm pointing now, and then there's this halo where some people had damage further back, some people had damage further anterior. But now we can look at the behavior of all these individuals and see what they have in common and relate it to the part of the brain that was damaged in that individual. Now, in a neurolaw context, typically we're dealing with one individual who's going to have their particular brain with its particular idiosyncrasies. And so we need to understand the problem of relating individuals to group averages. This illustrates the method of transcranial magnetic stimulation. The device is an electronic coil shaped like a figure 8. And when an electric current flows, a magnetic field is induced. You don't need to know these equations. I just want to illustrate Maxwell's equations that articulate the relationship between electrical current flow and magnetic field generation. And so in the upper right, we see when the current flows in the coil, a magnetic field is induced. That magnetic field can propagate through the skull, through the skin and skull into the brain. And that magnetic field, in turn, causes electric current to flow to change the state of neurons in a discrete part of the brain. So TMS over primary motor cortex in the precentral gyrus can cause twitches of the hand or the face, depending on where the coil is located. TMS over visual cortex in the back of the brain can cause the appearance of flashes of light. TMS over dorsolateral prefrontal cortex can change how you judge things or choose. It's a non-invasive technique, though, so its spatial resolution is not very high, and it only penetrates so deep into the brain. I want to illustrate another technique of direct electrical stimulation of the brain. This must be done invasively. This slide portrays the treatment of Parkinson's disease. And so there's the lead or electrode that's placed in this particular region of the basal ganglia known as the subthalamic nucleus. That's placed under surgery and located correctly. And then the electricity needed to generate the current to stimulate the subthalamic nucleus is generated from a pulse generator that's implanted in the chest, something like cardiac pacemakers and so on. And so then there's a wire connecting it together. And so now the individual can tune the frequency of the stimulation to treat the symptoms so that their movement disorder symptoms are alleviated. It's possible to electrically stimulate any part of the brain, and as researchers with animal models, this is one of the characteristic ways of investigating the function of different parts of the brain. In humans, electrical stimulation like this is used commonly to treat movement disorders like Parkinson's disease. There's experimental work using electrical stimulation of ventromedial frontal cortex to treat depression. The last, and in some sense the most common way to manipulate the brain is through psychoactive substances like drugs, for example, caffeine and other drugs of abuse. And so this slide is a diagram of a synapse. So the axons at the top coming down to form the synaptic cleft. On the bottom is the postsynaptic region of the dendrite of the postsynaptic cell. We see the vesicles with the blue neurotransmitters in them, and This is illustrating the site of action of different drugs, and we'll walk through it carefully. On the left, we're illustrating the example of agonist drugs, and on the right, antagonist drugs. So an agonist is a drug that, when delivered, acts as if it's the neurotransmitter. It potentiates the action of the neurotransmitter. An antagonist blocks the effect of the neurotransmitter. Let's work our way through this slide one box at a time. So in the upper left, an agonist drug can be a drug that increases the synthesis of the neurotransmitter molecule. So if I put more neurotransmitter in the vesicle, or I make more vesicles, each action potential that comes down and releases a neurotransmitter will release more neurotransmitter. So it will have more net effect on the postsynaptic membrane. The next approach is a drug that increases the number of neurotransmitter molecules by destroying the degrading enzymes. So when the neurotransmitters are built, there's other enzymes that will tear them down. By blocking the mechanism that tears them down, you keep more of them around to release into the synaptic cleft. We can also have a drug that increases the release of the neurotransmitter molecules in the terminal. All right? So the process of the vesicle binding and releasing the neurotransmitter is itself, like all processes, error-prone. And so drugs that allow that to be more efficient and release more neurotransmitter will have an agonist effect. Other drugs can bind to what are known as autoreceptors and block their inhibitory effect on neurotransmitter release. So synapses are highly regulated mechanisms. And I didn't tell you this before, but when a neurotransmitter is released, there's receptors in the postsynaptic membrane to depolarize or hyperpolarize the membrane and so on. But there's also receptors on the presynaptic axon assessing how much neurotransmitter is being released. If too much is being released, then that autoreceptor can have influence to prevent or reduce the amount of neurotransmitter released to optimize the system. So if we prevent the autoreceptor, where am I? If we prevent this autoreceptor from inhibiting the release of the neurotransmitter, then more neurotransmitter will be released. There's layer upon layer of regulation, and drugs can operate at each of those layers. Next, and in the simplest sense, here's the simplest sense of an agonist is a drug that binds to the postsynaptic receptor and activates it. It's not the key for the lock, but it's a fake key that the lock treats as if it were the original key. And finally, the last agonist we'll consider, the last agonist mechanism is a drug that blocks the deactivation of the neurotransmitter. All right? It can block the reuptake, or it can block the degradation of the neurotransmitter in the synaptic cleft, and that will result in more net effect. Now let's turn to the antagonistic drugs. So the neurotransmitter has to be synthesized and then packaged in the vesicle to be released. And one method of antagonist drug action is blocking the synthesis of the neurotransmitter in the first place. So if you have less neurotransmitter available, less can be released, so it'll have less net effect. Another antagonistic drug effect is causing the neurotransmitter molecules to leak from the vesicles and be degraded by enzymes outside the vesicles. A third antagonistic drug effect is to block the release. So when the vesicles bind, they may get stuck so that they don't release into the synaptic cleft. A fourth antagonistic drug effect is to activate the autoreceptors so that the synapse thinks, or the axon thinks it's putting too much neurotransmitter out, so it down-regulates itself. And the fifth and clearest sense of an antagonistic drug effect is a drug that binds to the receptor, preventing the the neurotransmitter from binding, but not having the effect. It jams the lock. These are some of the complex mechanisms of drugs that affect the nervous system and synaptic transmission. So you should go away with an appreciation for how complex synaptic transmission is, how many ways drugs can affect it, and also how regulated synaptic transmission is intrinsically. What that means by implication is drugs have side effects. Any influence it has on one aspect of synaptic transmission can be counteracted by all these other mechanisms of synaptic transmission.